Okay, so this is our final lesson on different modes of inheritance, and today we're going to tick off the final two, which is codominance and multiple alleles. So to start us off with codominance, codominance is a specific scenario where two or more alleles are equally dominant to one another. So previously we've been looking at dominant versus recessive alleles, but in the case of sickle cell anemia, we see two alleles with both being observed in the phenotype. So sickle cell anemia is due to a variation of hemoglobin where our red blood cells end up looking like this sickle cell on the right-hand side of my screen. This is in comparison to my normal red blood cell on the left-hand side, which has that biconcave disc structure. So what is this caused by? If we have a look at our alleles for sickle cell anemia, our normal shaped hemoglobin is thanks to this HbA. Hb, in this instance, standing for our hemoglobin. Now in the case of HbA, we can also represent this as Hb with a superscript, capital A. This is for our normal shaped hemoglobin. The abnormal hemoglobin, in this case for sickle cell, is represented by our hemoglobin and a superscript S. Now these two particular alleles are equal to one another. They are codominant. Now the abnormal hemoglobin, being sickle-shaped, results in less oxygen being transported by erythrocytes. So sickle cell anemia in individuals who are homozygous for the sickle cell trait, it can be lethal and often is. If an individual, however, is heterozygous, they have both types of these cells. So when we look at the particular genotypes of individuals, an individual who is normal red blood cells, we can represent as HbA for the first allele, and for their second allele, HbA once more. So this is a homozygous dominant for normal red blood cells. In those who are heterozygous for the sickle cell trait, they have both a copy of the normal allele and also a copy of the sickle cell allele. Now, as a consequence of them having both, neither gene in particular dominates over the other. And these individuals usually survive because they have enough red blood cells to supply their tissues with oxygen. So it's only where we might have a lower oxygen level, so maybe hiking Mount Everest, for example, where we have lower oxygen levels, that someone with a sickle cell trait might be in risk of some danger. Finally, as we've mentioned previously, those with sickle cell anemia are homozygous for that sickle cell trait. And in this instance, unfortunately, it is usually lethal because these sickle red blood cells just do not have the capacity to carry enough oxygen around to body tissues. So now that we've had a look at codominance, this is somewhat expanded on in our example, but we're going to have a look now at multiple alleles, and the example being the ABO blood system. So multiple alleles are where we have more than two alleles for a particular gene or for a particular characteristic. So these ABO blood groups are great because they show us three particular alternative alleles. We can have an A, a dominant B, or we can have our recessive allele as well. So when we have a look at the particular blood types, we can see that these three alleles present us with four possible blood types. Our first blood type A is where we have the antigen A expressed on the surface of the red blood cell. In the genotype, this can be represented by two particular variants, the first of which is a homozygous dominant for the A allele. The second is heterozygote, where the A allele masks the recessive allele here. When we get to type B blood now, type B has the B antigen on the surface of the red blood cells. It mirrors very much what happens with our A antigen, where we can have a homozygous dominant individual or we can have a heterozygote. Now, to take us back to codominance just for a second, we can also have type AB blood. And in type AB blood, we have the A antigen, and we also have a copy of the B antigen. So both are expressed on the surface of red blood cells. And this is because neither the A nor the B is dominant over the other. Finally, we can also have type O blood. And in type O blood, neither antigen is presented, and instead we have 2 copies of the recessive allele. And it's clear to see when we look at our lowercase i's here that there is no A antigen in the genotype, nor is there any type B, so the red blood cells themselves have no antigen presented. So to recap now our 4 models of inheritance that we've looked at. In our first lesson, we looked at autosomal inheritance. We looked at using uppercase letters for dominant alleles and lowercase for recessive alleles, and we could pick from a variety of letters. In our second lesson, we looked at sex-linked traits, and we looked at how when we're writing these genotypes, we need to make sure that they are expressed using the X and Y chromosomes. In this lesson, we have just looked at codominance, and the example for codominance that we have looked at specifically has been sickle cell anemia. In sickle cell, we looked at normal individuals with regular red blood cells having the genotype of HBA, and those affected by sickle cell having the allele HBS. Our last example here today has been looking at our multiple allele system, and the example that we've spoken about has been the ABO blood system, where we can have type A blood, we can have our B allele, and we can also have a recessive allele. And this can lead to a 4th combination where we can be a codominant between type A and B blood, leading to type AB blood.